In the vast and complex world of tropical medicine, some of our oldest enemies are hiding where we least expect them. Leishmaniasis remains one of the most common tropical diseases worldwide, affecting millions across the globe. Welcome to a deep dive into a groundbreaking study published in the American Journal of Pathology and Research. Authored by Dr. Ofelia Perez Olvera from the Faculty of Medicine at UNAM, Mexico, this research challenges what we know about the Leishmania life cycle. To understand this discovery, we must first look at the disease itself. There are three main forms of Leishmaniasis. Visceral Leishmaniasis is the most severe, often targeting the liver and spleen. Then there is cutaneous, the most common form, which generally causes distinctive skin ulcers. And finally, the mucocutaneous form, which affects the mucous membranes of the nose, mouth, and throat. Despite years of study, the exact histopathological nature of the lesions formed during infection remained a mystery. To solve this, Dr. Perez Olvera's team reproduced the disease in a controlled murine model. They inoculated the plantar pad of BALB/C mice with the species Leishmania mexicana. The goal was to define and identify the histopathological nature of this specific murine model. The team utilized advanced histological, immunohistochemical, and immunofluorescence techniques to describe the tissue in detail. What they discovered was unexpected. The lesion at the site of infection was actually a lipoma. This means that adipose tissue, or body fat, is a primary and vital component of the lesion. Even more significant, this adipose tissue was populated by Leishmania mexicana in its amastigote form. Adipose tissue is typically known for its metabolic functions, primarily energy storage. This leads us to recognize the active role fat plays in the reproduction and migration of the parasite. To reinforce these findings, the researchers conducted a second study by inoculating the parasite at the base of the tail. The results were consistent. Both models showed the same morphological description where adipose tissue is a dominant element. Based on these findings, the research team suggests a fundamental update to our understanding of the disease. They propose that adipose tissue should be formally added to the Leishmania species life cycle. This is not just a theoretical update. With over 12 million people infected worldwide, understanding how the parasite survives is critical. Adipose tissue may act as a sanctuary, protecting the parasite from immune responses and pharmacological treatments. This helps explain why Leishmania can persist in the body for long periods, often leading to treatment relapses. The metabolic relationship between the parasite and host fat cells is a dynamic and active exchange. The parasite utilizes these energy reservoirs to fuel its invasion, proliferation, and eventual migration. In Mexico, where the disease is endemic in 13 states, these findings have immediate relevance for public health. Managing the localized cutaneous form, often found in coastal regions, requires this deeper understanding. Future therapeutic strategies may now need to account for these fatty reservoirs to achieve total clearance. Targeting the parasite's ability to utilize adipose tissue could lead to more effective treatments. This research marks a significant shift in tropical medicine, moving beyond the traditional focus on macrophages. By identifying the specific histopathological nature of the Leishmanial lesion, we see the parasite's survival strategy in a new light. The work at UNAM's Faculty of Medicine continues to push the boundaries of what we understand about infectious diseases. This multi-institutional effort highlights the importance of collaborative research in solving global health challenges. As we continue to explore the hidden sanctuaries of pathogens, we move closer to a world without the burden of neglected tropical diseases. This discovery by Dr. Perez Olvera and her team is a testament to the power of persistent, detailed morphological study. Thank you for joining us as we explore the future of pathology and infectious disease research. Stay curious and keep supporting the research that changes lives every day. The American Journal of Pathology and Research, bridging the gap between discovery and care.